Hi everyone, this is Dr. Karen's, and today I'm going to talk a little bit about the Kirby Bauer test as part of our lab assignment for microbiology. So another name for this test is the diffusion susceptibility test. And the whole purpose of this test is basically to figure out which antibiotic drugs work the best against different bacterial species. So which drug will work best on that bacteria? Another way to think about this is, if you think about it in terms of the bacteria, is the bacteria susceptible to this drug or another drug? Or is it resistant to these drugs or some of these drugs? And the reason for wanting to know this is really in the hospital setting. So this test is still being done today on patients who have bacterial infections that might not be responding to typical antibiotic treatment. And so you might need to find out, well, is there a better drug for this infection that I could be using instead? And so you do this Kirby-Bauer test and figure out which antibiotic works the best against that particular infection. So some terms that I need to clarify here, because in the past some students get mixed up in terms of what we're actually talking about. So these are terms that are describing bacteria in the environment of a drug. So susceptible, what does that mean? It means that a bacteria is sensitive to an antibiotic, meaning that that drug can kill the bacteria or inhibit its growth. And that means the drug will be an effective treatment for that particular bacterial infection. Another term is resistant, and that means that the bacteria is somehow immune to that particular antibiotic or whatever drug you're testing on it. And that means the drug can't kill the bacteria or inhibit its growth. And therefore, that drug will not be an effective treatment to treat an infection caused by that bacteria. Now, how do bacteria become resistant to different drugs? If you go back to Chapter 7 in this class, you will remember that bacteria usually acquire resistance in a couple of different ways. They could just acquire a random mutation that makes them resistant to that particular drug, or they can kind of inherit a resistance plasmid from another neighboring bacteria via conjugation. And then sometimes a bacterial response to a drug will be somewhere in between susceptible and resistant, and we use the term intermediate to describe that. So in other words, the drug kind of works against that bacteria, but not very well. And in the hospital setting, you might want to look for better options to treat this infection. The takeaway from this, and what I want to emphasize, is that it is the bacteria that are either susceptible, resistant, or intermediate to different antibiotics. It's not the other way around. Drugs cannot be resistant to things. If you are resistant or susceptible to something, you have to be living. And that's what we're talking about here. So use these terms to describe how the bacteria responds to the different antibiotics. Okay, so this Kirby-Bauer test, remember what we're trying to do is test a variety of different antibiotics or antibacterial drugs and see which ones work well against a particular bacteria, which ones are going to kill the bacteria, which ones are not going to work very well. So the basic procedure is, first, you need the bacteria, and we use petri dishes or plates for this experiment. You need to completely cover the surface of the plate with the bacteria. You will not use an inoculation loop for this. You will need a liquid culture of a bacteria to begin with, and instead of using an inoculating loop, you're going to use a swab, a sterile swab, which is basically like a giant Q-tip. So you, using aseptic technique, you take that swab, you dip it in your liquid culture, and then you spread it all over the plate. And you can kind of see in this picture here, if you're looking at the background of the plate, you can see that every inch of this plate is covered with bacteria. You will not see the bacteria until you incubate the plate. So you spread the bacteria all over the plate, and then you take these prepared little discs of paper that have been saturated with different antibiotics, and you usually using tweezers, you will gently place a disc on the plate. If the plate is big enough, if you're using a standard size plate like the one in this picture, you can put more than one disc on the plate. Usually, I would say between 5 and 6 discs. You want to have enough space around each of the discs in order to read your results accurately. So we have a plate that has been covered with bacteria and now has various discs of antibiotics placed on the surface of that plate. And now we incubate the plate. So what's going to happen? Ideally, the antibiotics that are on these discs are going to start diffusing through the agar. The bacteria that you spread on the plate is going to try and grow. And the idea is if the antibiotics that are diffusing into the agar can kill the particular bacteria that you put on the plate, then that antibiotic will prevent the growth of the bacteria around the disc. And it's this clearing here that you see in this picture. That is what we call a zone of inhibition. And you see these clear rings around some of these antibiotic discs. When you see clear, that means there's no growth there. And that's because the antibiotics diffused through the plate and prevented bacterial growth. What you will do in order to make some conclusions in this experiment, you need to measure the zone. And so here is how you would measure that zone. You want to measure the diameter starting from one end of the zone straight across, straight through the disc to the other end of the zone. That's what you want to measure. So we're trying to figure out which antibiotics worked the best. So in this picture, all the discs are of different antibiotics, and you can see that some of the zones are large, some of the zones are smaller, and some antibiotics didn't produce a zone at all. At first glance, it seems logical to assume that if there's a bigger zone of inhibition around an antibiotic disc, well, then that must mean that that antibiotic worked really, really well and worked better than other antibiotics that have smaller zones. And then if you don't see a zone at all, it means that antibiotic did not work at all. It did not kill the bacteria. So that's a very logical conclusion to make, but it is not accurate because you cannot just look at the sizes of the zones of inhibition and make conclusions about the strength or the effectiveness of that antibiotic, because antibiotics will diffuse through the agar at different rates. So if you look at this particular zone of inhibition, it's a lot smaller than the one to the left of it. At first glance, you might think, oh, that must mean this antibiotic didn't work very well. But like I said, it's not necessarily true. It could just mean that this antibiotic diffused through the agar more slowly. And so it didn't have enough time to kill as much bacteria as another antibiotic. So you can't just look at the size of the zone of inhibition alone to determine how well the drug worked. You have to take into account the different diffusion rates of the different antibiotics. How do you do that? There are standardized tables that are already published where somebody kind of figured out at what rate do these different antibiotics work. And so once you've incubated your plates and you take them out of incubation and you can see your zones and you measure the diameters of those zones, you have to compare each of those diameters to a standardized table for each of the antibiotics. So let me explain how you would do that. So this last slide here is an example experiment. So the table on the left is giving you results of a Kirby Bauer test that was performed on bacteria A. It doesn't matter what the bacteria is. 3 different antibiotics were used: penicillin, novobiocin, gentamicin, and the zone of inhibition for each of those antibiotics was measured. Notice that the zones are all the same size. If you're looking at this data, it is very easy to just say, oh, the zones for all 3 antibiotics are exactly the same size. That means all 3 antibiotics worked equally well to kill bacteria A. But that is not true because, like I said earlier, you have to account for the fact that these 3 different antibiotics diffuse through the agar on the plate at different rates. So you need to compare these results with the standard table, and that's what the table here is on the right. This is a standard reference chart for these particular antibiotics, and you'll see that there are different ranges of zones of inhibition, and depending on which range your measured zone in your experiment falls under, that will tell you if bacteria A is susceptible to the drug, intermediate, or resistant to the drug. So let's go through this one by one. So if we're looking at penicillin, I'm looking at my results of the test. Penicillin gave a zone of inhibition of 16 millimeters on bacteria A. I take this 16 millimeters and I go and compare it to the reference chart. So for penicillin, a zone of inhibition of 16 millimeters falls in this intermediate range. Intermediate means, for penicillin, means 12 to 21. 16 falls within that range. Then now let's look to novobiocin. The zone of inhibition is 16 millimeters. What does that mean for novobiocin? For novobiocin, 16 falls under the resistant category, because for novobiocin, a zone of 17 millimeters or less means resistance. And then last, let's look at gentamicin. 16 millimeter zone of inhibition. For gentamicin, that means susceptible. Susceptible is a 15 millimeter zone of inhibition or more. So what exactly, how do you make the conclusion, and what words do you actually say if someone's asking you, how do you interpret these test results? So you do what I just did. You look at your test results, you compare the zones that you measured to the reference chart. Penicillin, a zone of 16 means intermediate. Well, what is intermediate? That means that bacteria A has an intermediate effect to penicillin, meaning it's not super susceptible, but it's not resistant either. It's somewhere in between. For novobiocin, 16 millimeter zone of inhibition falls in the resistant category. That means that bacteria A is resistant to novobiocin, meaning novobiocin doesn't work. Even though there's a zone for that drug, the zone is not significant enough to mean that the drug actually worked. And then finally, gentamicin, 16 millimeter zone of inhibition, that puts it in the susceptible category, puts the bacteria in the susceptible category. So that means bacteria A is susceptible or sensitive to gentamicin. So which of these drugs works the best against bacteria A? Gentamicin, because the zone that you measured placed the bacteria in the susceptible category. The drug successfully killed the bacteria. Novobiocin didn't work at all because the bacteria falls in that resistant category due to the size of the zone. So it's really important that you understand that you can't just look at the size of the zone. And in this example in particular, I purposefully picked 16 millimeter zones of inhibition for our test to show you that even though these zones are exactly the same size, they mean very different things for each of the drugs once you compare those zones to the standard reference chart. So when you're doing this lab, the setup is very easy as I described. Measuring the diameters is fairly easy. It's just a ruler. The tricky part here is understanding how to use a reference chart and then understanding what those things mean. So let me just reiterate. You want to know which antibiotics worked and which ones didn't. And another way to say that is which antibiotics is your bacteria susceptible to, which antibiotics is your bacteria resistant to, and which antibiotics fall somewhere in between. And again, to reiterate, this test continues to be done in hospitals today in order to determine the best plan of attack against different types of bacterial infections.